Here is a short video abstract describing our work on the combined effects of oxalic acid sublimation and brood breaks on Varroa mite and deformed wing virus levels in honeybee colonies. Annual losses of honeybee colonies are often attributable to the ectoparasitic mite Varroa destructor and associated pathogens such as deformed wing virus. In this study, we investigated the effects of two methods of Varroa mite control, oxalic acid sublimation and natural swarming on colony health and productivity. We monitored the colonies over the course of one season in central Pennsylvania, from April through October, assessing Varroa mite drops, colony weights, and honey yields. In the spring, many beekeepers rely on packaged bees to replace lost colonies or start new ones. However, these packages often come with high levels of mites and are treated with miticides upon installation. Oxalic acid is one of the most common treatments for Varroa mite control among beekeepers. Our previous study showed that sublimation was the most effective method to administer oxalic acid during broodless periods. We installed 27 packaged bees in April, of which 14 were treated shortly thereafter with oxalic acid sublimation. Swarming occurs when the queen and the majority of workers leave the original colony en masse to establish a new colony elsewhere, for example, a tree cavity. Colony swarming is a natural behavior that can also control mite numbers through a break in brood production that leads to a break in mite reproduction. In this study, colonies were allowed to swarm over the course of the study to assess the role of natural mechanisms of mite control on colony health. Our results suggest that mite drops increased immediately after the oxalic acid treatment in the spring. However, mite populations recovered, leading to high mite levels in the fall in both treated and untreated colonies. We also observed marginally significant reductions in honey yields among colonies that were treated. To assess swarming effects, we compared mite levels throughout the season among colonies which swarmed in early summer, May and June, with colonies that swarmed in July and August. We found that late summer swarming was correlated with lower levels of Varroa mites and deformed wing virus in the fall, suggesting that implementing a brood break in the midst of the summer could play an important role in reducing mite populations and deformed wing virus levels for colonies originating from packages. Additionally, oxalic acid-treated colonies that swarmed had significantly lower mite levels. Similar with treated colonies, we also found a slight reduction in honey yields among colonies that swarmed in the summer. However, beekeepers may be able to avoid significant honey losses by dividing colonies after peak nectar flow in the summer. Overall, our data suggest that oxalic acid applications to packaged bees in the spring, in combination with summer brood breaks, had a significant effect on the reduction of mite levels in the fall, when colonies were preparing to enter winter. To mimic the beneficial effects of swarming, beekeepers can make artificial swarms through splitting or colony division. These methods can easily be implemented into an integrated pest management program.